Great. Hello. Good morning or good afternoon, depending where you are. I'm really excited to be here. Thank you so much for the invitation. Yeah, I don't need much introduction after Suzanne's talk. I'll focus on the link between brain-to-brain synchrony and classroom learning, specifically learning in small groups of students. So just to summarize some of the studies that Suzanne already described that we've done in classrooms, in the first study that was published in 2017, we followed a group of 12 high school biology students throughout an entire semester, and were able to demonstrate that the degree of synchrony between students is predictive of how engaged students are. Were students who were more engaged in activities like lectures and watching instructional videos showed higher synchrony with their peers. And brain synchrony was also predictive of social closeness between students, where pairs of students who reported feeling closer to one another showed higher synchrony. And that also relates nicely to the talk that we heard yesterday from Caroline Parkinson, sorry. And then in the follow-up study that was published more recently with Dana Bevilacqua and others, we extended this framework to student-to-teacher synchrony. So here we measured not only the students, but also the teacher. Clearly, this is more challenging because the teacher is speaking and moving, and that introduces noise to the EEG data. But nonetheless, we were able to show that student-to-teacher brain synchrony is also predictive of student engagement and social closeness. Here, social closeness is the degree of closeness between students and teachers. However, one big question that remains is, what about learning? So do students learn better when their brains are in sync? And that's the main question that I'll address today. And the reason that this question is important is because there are some mixed results regarding learning outcomes and brain synchrony in the literature. So in the Bevilacqua et al. Study, we actually did not observe a significant link between either student-to-student or student-to-teacher brain synchrony and learning outcomes. So the students were given a quiz at the end of each lesson, and there was no significant link between their performance on the quiz and their brain synchrony. However, in another study by Cohen et al., mind you that was not a classroom-based study, but rather students watching instructional videos individually. And over there, there seemed to be a significant association between synchrony across students and their performance on a test. So we found this discrepancy to be intriguing, and we decided to investigate it a bit further in a study that we recently conducted with Emma Laurent and Henry Valk, where we measured small groups of 4 students in a simulated laboratory classroom. Overall, we had a larger sample of 31 students, larger than what we've used before in classrooms. The students here were not high school students, but rather college students. These were primarily psych students, and the content that they were taught was biology or chemistry related. So they had very limited prior knowledge about this topic. We wanted to assess their learning very systematically. So we brought them in the lab a week before the EEG session to take a pretest. And then during the EEG session, they went through a sequence of 4 mini lectures, again, on different topics in biology and chemistry. And immediately after each lecture, they took the same test that was administered the week before. And the same test was also administered a week later to look at delayed retention. So here are the behavioral results. So we first wanted to make sure that students indeed learned from these mini lectures, and it seems that they had. So as I mentioned, students had very limited knowledge about this topic. So at the pretest, they scored rather poorly at around 40%. And then immediately after each lecture, there was a highly significant increase in their content knowledge to around 70–75%, which for the most part sustained a week later with some decline, which is expected. But the main measure that we were interested at was the difference between the delayed post-test and the pre-test. And this delta is what we call learning gains. And the reason that we focus on the delayed post-test is that in education, we want to generate changes that sustain over time, right? We want students to do well, not only in an immediate test, but also be able to use their knowledge over time. So the question here is, can we predict learning gains based on brain synchrony across students? But we also considered other potential predictors. We looked at self-reported engagement. So immediately after each lecture, we also asked students to rate how engaged they were during the lecture on a scale from 1 to 7, where 7 was highly engaged and 1 was not engaged at all. So we had this self-reported engagement that we also included in previous studies. Another measure that we looked at was intra-brain synchrony. So we used the same across-brain synchrony, which is circular correlation in this case, to measure intra-brain synchrony. So by intra-brain synchrony, I mean the synchrony across different EEG electrodes within an individual brain. And the reason that this predictor is interesting is because we don't yet know what is the added value of looking across brains compared to just looking at individual brains. So we wanted to see whether learning can be better predicted by looking across brains compared to looking just at individual brain measures. So these are the 3 predictors that we looked at: self-reported engagement, intra-brain synchrony, and across-brain synchrony. And while I take a sip of water, I would like you to think, what pattern of results would you predict? So which one of these 3, of course there can be more than one, would be a significant predictor of learning gains in this study? Okay, so here are the results. So in terms of self-reported engagement, based on prior research, we hypothesized that how engaged students are during a lecture should predict how well they learn, but surprisingly, there was only a non-significant trend in the data. And that raises questions regarding metacognition, regarding how aware or how accurately students can report their level of engagement, and I'm happy to discuss that later. With intra-brain synchrony, there was no significant association whatsoever with learning gains. However, brain-to-brain synchrony across students highly significantly predicted to what extent students retained information over time, where students who showed stronger synchrony with other students in the room also learned more information and were able to retain this information over time. And we included these 3 predictors in a single multi-level model, and were able to show that brain-to-brain synchrony predicted learning above and beyond what can be learned from self-reported engagement and individual brain measures. We also looked at, in addition to intra-brain synchrony, we looked at other measures like overall power in different bands of the EEG signal, and the results were the same, where brain synchrony was the only significant predictor of learning under these circumstances. I should also say that these results correspond to the alpha band, that is EEG activity in the 8 to 12 hertz band. But we also looked at other frequency bands, and alpha band was the only frequency band that significantly predicted learning, again, looking at brain-to-brain synchrony. And we can explain that in different ways. One potential explanation is that it has to do with signal to noise. Alpha, as many of you I'm sure know, is the strongest signal in EEG. So that might be one reason. Another reason is that alpha has been associated across many studies in the past with attention. As Suzanne already mentioned, we think that brain-to-brain synchrony might capture variation in shared attention. And when students pay attention and show shared attention with others, perhaps they can also learn information better. So that might be another explanation to this pattern of findings. But we wanted to take that a step further. So far, I only talked about brain synchrony across the entire duration of, let's say, a 7-minute lecture. But can we say anything about the variation of brain synchrony within a lecture? So as we know, EEG has good temporal resolution. Can we extract any meaningful information about how brain synchrony unfolds over time? So what I didn't tell you yet is that in this post-test that we had immediately after each lecture, as well as a week before and a week after, there were 10 multiple choice questions per topic. And each question corresponded to a different segment of the lecture. So what we did was, for each test question, we went back to the lecture transcript and identify the specific time window at which the teacher provided critical information to answer that question. So for example, this question was about bipedalism and adaptation to different ecosystems. So we were able to identify the specific segment of the lecture, in this case between these time points, where the teacher provided the critical piece of information that students needed to answer that question. Now, since these lectures were given live to multiple groups of students, we had to repeat this process for each group of students. The lectures were consistent, but of course they were not produced the same way each time. But regardless, for each test question, we identified a specific segment of the lecture, and now we could compute brain synchrony rather than throughout the entire lecture. We could compute brain synchrony specifically for this time window, as well as the time windows of the other test questions. And the finding here was that alpha band brain-to-brain synchrony was significantly higher for questions where students showed learning, that is, questions that they answered incorrectly in the pre-test and correctly in the post-test, compared to questions where students did not demonstrate learning, that is, questions where their answers did not change between the pre and the post-test. And I think that this finding is important because it suggests that brain synchrony can also provide us with temporal information and be used to make specific predictions about how students retain information from, in this case, lectures, in the future, perhaps from other teaching activities as well. So far, I only focused on student-to-student brain synchrony, but you might wonder about student-to-teacher brain synchrony and its linkage with learning. Since the teacher here was the speaker and the students only listened to these lectures, we predicted, based on consistent with prior research, that perhaps the teacher's brain activity would precede students' brain activity. Just due to the fact that, again, the teacher was the speaker and the students were the listeners. However, we also considered the opposite. So we considered time lags where students' brain activity would precede the teacher, perhaps trying to make predictions of what the teacher is about to say. And we know that this process of anticipation, of making predictions, is really important in human communication. Probably as I'm speaking, you're already making predictions about what I'm about to say or show you in the next slide. So for each time lag between the student and the teacher, we recomputed student-to-teacher synchrony and correlated it with learning gains. And that's what I'm showing you here. So this graph shows the correlation between student-to-teacher synchrony and learning gains as a function of the temporal lag between students and teachers. So negative values correspond to the teacher preceding the student, and positive values correspond to the student preceding the teacher. And as you can see, student-to-teacher brain synchrony best predicted learning when adjusting for a roughly 200 millisecond time lag between students' brain activity and teachers' brain activity. And that, if you think about the time course that it takes our brain to process language, or speech, that makes sense, that it took the students perhaps 200 milliseconds or so to align with the teacher's brain activity in order to best predict learning. So that was the case overall across all 32 EEG channels that we had in this study, but we also wanted to look at how this effect perhaps varies across different locations. And in this topographical map, each EEG electrode is color-coded based on the temporal lag between the student and the teacher that produce the best prediction of student learning. So blue corresponds again to teacher preceding the student, and yellow correspond to student preceding the teacher. And you see that most electrodes are blue. As I just showed you, overall, the teacher's brain activity came in first in order to produce the best prediction of learning. However, you can see that intriguingly, central and frontal electrodes show the opposite pattern, where the correlation between student-to-teacher synchrony and learning peaked when the student's brain activity preceded the teacher. And this is actually consistent with some work from Uri Hassan's lab on speaker-listener brain coupling or brain synchrony, where frontal cortex was shown to perhaps engage in this process of generating predictions about what the speaker is about to say. And synchrony in these frontal regions was found to best predict comprehension of a story that people were listening to during a functional MRI brain scan. So in the remaining time, I'd like to very briefly tell you about where we're going to take this research next. So in this study, I focused on lectures, but as an education researcher, I care a lot about active learning and about how students learn collaboratively in groups. And the reason that this question is important is that we know based on prior research that overall collaborative learning is more effective than individual learning, but we don't really have a good understanding yet about how and why collaborative learning works. And furthermore, we know that sometimes collaborative learning can be ineffective. Sometimes even highly capable individuals, when they're placed in groups, they fail to work together effectively. And the question of why do some groups succeed and some fail is really important both within education as well as other fields. And I feel that this notion of brain synchrony can provide some important insights about collaborative learning in classrooms. So to start asking questions about that, we conducted another study that was clearly pre-COVID, and hence the lack of social distance between the students. So here we collected EEG data from 4 high school students, and unlike the previous study that I described, the students were not passively listening to a lecture or watching a video, but rather engage in a group task. In this case, they were given different materials and were asked to construct a model of a cell. And I'm going to show you a very brief video just to illustrate how that experiment looked like.
So, oh my God, we fully have to
This is the endoplasmic reticulum, these are the ribosomes. This is a nuclear membrane, this is a nucleus.
Bio is going to be next year. Oh my God.
Wait.
Yeah, I know, I'm doing that for a reason.
Yeah, so as you can tell, the students were speaking to one another, they were manipulating objects. Quite interestingly, in this video specifically, the two female students were clearly in the lead and the two male students seemed less engaged. Many interesting questions to look at. It's a very rich data set. We have video data. We transcribed all these conversations between students. This is still ongoing. I don't have brain synchrony data to share with you just yet, but very briefly, we already coded all these videos, and interestingly, at about 15% of the time, the students seem to be idle, so seem to be not engaged in the task, at least from the outside. And clearly with EEG, we can also start asking questions about how cognitively engaged they were in this task, even when they seem to be disengaged. In about 12% of the time, 13%, they seem to be completely off task. And here's one example. So this is a transcript of a student conversation where they start by talking about the task. So they talk about endoplasmic reticulum and whether they should add ribosomes to their figure. But then the student here, made a comment about how tired she is, and that really derails the conversation. And this is an interesting moment in time because it reflects a switch between on-task and off-task interaction. Perhaps this switch can explain why this group was more or less effective in completing the task. And the next step here would be to incorporate the brain synchrony data. And as I hinted before, we hope to not only look at brain synchrony overall across the entire duration of this activity, but also look at a temporal variation, at how brain synchrony unfolds over time. One prediction based on our previous research is that perhaps moments of high brain synchrony would be characterized by high joint engagement of the students, whereas moments of low synchrony would be characterized by low engagement of students. But this is exciting because it's a data-driven approach and we don't really know yet what we're going to find. So with that, I would like to thank everyone who were involved in these different studies, and also thank you for your participation. And I also want to say that I'm recruiting a PhD student for next year. So if you have any good candidates, please send them my way. And I'm happy to take any questions now.
Thank you so much for your talk. And yes, I would highly recommend anybody, if I didn't already have my hands full, I would be applying for your studentship because the work you're doing is obviously incredibly important for us all, so that we can make progress in how we educate one another. I have a host of questions as usual. I probably should invite people I'm less interested in their work because then I'd talk less. But I don't know where to start. I suppose one of the first questions that comes to mind is you make this distinction binary-wise between on and off task. And I'm just wondering, because I assume from a learning perspective that you actually need variation between on and off task. I don't think you learn necessarily when you're always on. So that's one of the questions I have, and maybe you can have a stab at that first.
Yeah, that's a fantastic question. And in fact, I just published an opinion paper about this topic with some colleagues in life sciences education. And yeah, this is definitely not a dichotomy, it's rather a continuum. And what I think makes it even more interesting is that even when students seem to be on task, they can be on task in different ways. They can be there physically on task by manipulating objects. They can be verbally on task by just speaking about the task, or they can be both. And clearly, identifying when students are on or off task is quite challenging. It's not a clear cut at all. There were some instances that were easily identifiable, like this moment in time where, AH declared that she's so tired, but in other moments, it's unclear to what extent students are on or off task. So that's a challenge from a methodological perspective, but it also, I think, adds to the richness of this data. And the hope is that now we can use not only video data and perhaps analysis of student conversations, but also start to use EEG data to gain insights into this complex transition between on and off task.
Perfect. We have a question from Travis Wiltshire, who I'll just springboard into. He says, really fascinating work. How do we go about deriving the meaning of interbrain synchrony? I know you mentioned this example of high engagement. Are there other types of synchronous states? And that's not my words, these are Travis's.
Yeah. I'm not sure I can fully answer this question. I don't think that we yet have a good understanding of the mechanism of synchrony. So we know that at the most basis level, it's probably generated by stimulus entrainment. So having a shared input is important. But based on the data that I just shared with you and Suzanne and others throughout this conference, it seems that attention plays an important role. It seems that social closeness plays an important role as well. So clearly it's a complex phenomena with multiple facets. And that's why I feel that this data-driven approach where we start from the EEG data and then go back to the video can be informative because we don't really have enough information to necessarily generate all these different hypotheses, but and then letting the data tell the story might be a good strategy. Of course, then we can use these to generate new predictions and test them in more controlled ways. But I think
it is the question, is it not? What does interbrain synchrony mean, right? That is the question.
That's $1 million question.
It is. There is another question, perhaps $1 million, on the YouTube link, and it says, this is from Hain-Huni Lee. In your previous study, I remember not significant differences in interbrain synchrony between the video to discussion condition, or both being high. How do you interpret the difference of the brain-to-brain sync and its driving factor?
Yeah, that's a great question. So I think that question refers to the study that Suzanne described earlier, the 2017 Kern Biology paper, where indeed synchrony across students was pretty much as high. At least there was no statistically significant difference between discussions and videos. However, this comparison is tricky because the input is so different, right? I just mentioned that stimulus entrainment probably plays an important role. And with video, where you have both visual and audio input, they're highly, they have very clear temporal structure. That can, by itself, can produce very high synchrony just due to bottom-up stimulation. So this comparison between discussions, which are more open-ended, and to videos, this comparison is tricky. I'm actually more interested now that we feel a bit more comfortable with this idea of bringing EEG into classrooms. As I mentioned, I'm more interested in focusing on discussions alone and asking questions about what is happening during discussions or other group activities.
So, but these transition moments and so on, which sort of brings us to a close, and where I will use this moment, first of all, to thank you, Ido, for your talk and for answering all our questions and inspiring us to try and better understand how we learn through interbrain coupling.